Chapter 2, which is on mitosis and meiosis. Excuse me. This should also be a review for you if you're in genetics, bio 3320. However, I would strongly employ you to go back and watch Chapter 1 and then venture into Chapter 2. These are some of the learning objectives that correspond to the sections in the textbook that we're using. And as always, I'd like to open up with a case study. So take a moment to pause the video to read this and then come back. Now that you have read this case study, I want you to think about how this concept of Hodgkin's disease affecting a male may differ from that of a female. And then to help you further understand more about the case study, I want you to ask yourself these questions. These are both genetics or scientific questions and some ethics questions that I think are or would be beneficial to you. And as you're learning this, while the context of the question centers around males, ask yourself, what would happen if this were a female? How would eggs be affected differently than males, and what would the long-term effects be? So just a brief introduction into this. In order to really understand how mitosis and meiosis fit together and their function into the grand scheme of an organism, you have to understand an organism and the components within it. So when we talk about an organism, whether it's the model organisms we were talking about in Chapter 1, they are composed of nucleic acid, i.e. DNA. Some can be RNA, but we're going to just say DNA. And this DNA is organized into chromosomes. That does not include viruses. But these chromosomes serve as vehicles for transmitting genetic information. In eukaryotic cells, the transmission of genetic material via the use of chromosomes involve mitosis and meiosis. Mitosis and meiosis are two major processes that involve genetic continuity of nucleic acid or nucleated cells. While there are many different processes that can be similar in ways in terms of the mechanism, the outcomes between the two are very different. So while these two processes have an overlap of processes within them, they're totally two completely different processes. For example, mitosis leads to the production of two daughter cells, each with the same number of chromosomes as their parent. Contrary, meiosis reduces the genetic content and the number of chromosomes by half. So the genetic information is reduced. And this is why oftentimes you hear meiosis referred to as reduction division. This reduction is essential if sexual reproduction is going to happen, because without doubling the genetic information with each new generation, it will cause genetic abnormalities, okay? And so understanding that process is extremely important. Also understanding the cells that participate in this. Your sex cells, your gametes, egg and sperm, which are haploid, are involved in or occur, they are part of the process of meiosis, where mitosis is for your somatic cells. Think of them as your body cells. Your non-sex cells, which are diploid, are going to engage in mitosis. One other thing that I want to point out is the structure of the chromatin, i.e. or chromosomes, is extremely important, is a clear indication of what's happening within the cell and what stage it's a part of. So looking at the chromosomes, the nucleic acid within the nucleus of the cell is a dead giveaway into identifying what state the cell is in either mitosis or meiosis and what is happening. So make sure you're paying attention to that. So let's look at the cell structure closely to help understand the genetic function that is occurring within the cell. And that is because the cell structure is closely tied to genetic function. There are two main types of cells, right? Prokaryotic cell, whether we're talking about bacteria or archaea, or eukaryotic cells, protists, fungi, plants, animal cells. They all share some common features, which is the plasma membrane, the presence of DNA, and ribosomes, which is a place where translation takes place. But they have some various differences. When you look at even this animal cell here, you can see that it tends to be much more complex than that of a prokaryotic cell that is shown here. Okay, much more simplistic, less complex. The eukaryotic cell, in this case, an animal cell, has membrane-bound organelles. It has, as you see, the nucleus, which contains the nucleolus, the nuclear envelope. It has mitochondria, it has the Golgi, it has the rough ER, the smooth ER, centrioles, free ribosomes. All these components are essential to an animal cell carrying out its function. The plasma membrane, which is present in all cells, is a partition that surrounds all cells. It helps to separate, or as your book says, delimits the external environment. So it separates the external from the internal portion of the cell. Plants have a plasma membrane, but they also have a cell wall, which is mainly composed of cellulose and polysaccharide. Bacterial cells have a peptidoglycan in their cell walls, and it also helps with identifying the cell type or the type of bacteria. The glycocalyx is another term for the cell coat, and it is the covering on the plasma membrane of animal cells, and it's made up of glycoproteins and polysaccharides. And these are important because they are identifiers for cells. Think of these as, hello, my name is badges. They help identify cells, and they serve as receptors on the outside of cells that are used for messaging each other, for stimulating responses, and a list of other factors. However, the function is to biochemically identify at the cell surface. And these receptor molecules pass signals. And what's interesting is when there is an abnormality in receptors, it can alter the cell function. It can also stimulate in, let's say, humans or animal cells, diseases and or disorders. The nucleus or nucleoid region in the bacterial cell is important because this is where nucleic acid is housed. A nucleus is found within the eukaryotic cell. A nucleus is not found in a prokaryotic cell. They have a nucleoid region, which is just a dense region in the cytoplasm that houses or where nucleic acid is located. The nucleus in a eukaryotic cell is membrane bound, so it has another membrane that allows for partitioning of the cytoplasm in the outside of the nucleus, and then the nucleoplasm, which is the liquid portion inside of the nucleus. And this allows for, again, partitioning and separation of the nucleic acid from other external factors. And as we get further into the semester and even beyond my class, and let's say you take molecular, you'll find that this is an integral part in the viability of a cell because it helps prevent enzymes that are specifically designed to degrade nucleic acid. The cytoplasm, which is the liquid portion of the cell, is found in all cells, and it is responsible for giving structure and fluidity to the cell. But this is where the cytoskeleton is composed, and the cytosol is that liquid portion of in the cytoplasm that helps with the surrounding organelles and their fluidity. You also have within the cytoskeleton, located in the cytoplasm, protein tubulin and microfilaments that help with the pulling apart or the separation of cells during that process of mitosis and meiosis. So the cytoplasm is extremely important as well. The endoplasmic reticulum. The endoplasmic reticulum is an important organelle because it is responsible for compartmentalizing, the cytoplasm, but it also helps with the synthesis of proteins. You will see that there are two parts, the smooth and the rough. The rough ER, or rough endoplasmic reticulum, is rough because of the presence of ribosomes, which is where translation takes place. The smooth ER appears smooth, and it has or is the site where you have fatty acid synthesis and phospholipid synthesis. So the rough ER is actually one of the most important organelles in the cell because this is where you have the production of proteins. Now, within the cytoplasm, you have other structures that also play a major role and have thought to exist within the eukaryotic cell due to symbiosis. where at one point the primitive mitochondria or chloroplast were thought to have been engulfed by early eukaryotic type cells, and now they live in a symbiotic and mutually beneficial relationship. But the mitochondria is the place where you have oxidative phases of respiration, ultimately where there are processes that occur, cellular processes that produce ATP, okay, and other byproducts. And then you also have the chloroplast, which is also the place where photosynthesis takes place, and other important factors. And these two are very important. In your more earlier or lower level classes, you learn that the mitochondria is known as the powerhouse of the cell, just referring to that because it is the site where ATP production is made. But that is such a simplistic example of what the mitochondria actually does. It's very important. It has its own nucleic acid. And you'll find that there are some genetic disorders that are tied directly to the mitochondria. The centrioles are important because they are found in the centrosome, and you'll find that the centrosome is important in animal plant cells because, again, this is the site where you have the organization of spindle fibers that help make up the microtubules that are responsible for the pulling apart of cells during mitosis and meiosis. And so these are extremely important areas in the cell that are essential for the processes that are necessary. Okay. So let's get into now a little bit more about the nucleic acid because we're going to start going into mitosis and meiosis. Homologous chromosomes. Homo means the same, okay? And so we see homologous chromosomes. There are chromosomes that are similar or the same. They may not be identical, but they're similar. An example of this would be chromosome 1, right? So you can have chromosome 1, but you have two versions of chromosome 1 because you got one version from your mother and one version from your father. While they house the same genes, the versions or the variants of that gene may differ. As we learned in chapter 1, that we get the formation of alleles because of mutations that evolve over time and that are incorporated, producing alternate versions of a gene. And this is how you get the production of homologous chromosomes. However, these chromosomes carry genes for the same inherited characteristics. So it may be your parent has on their chromosome one color for eyes, and then your other parent has another gene or allele for another eye trait. Okay, those are called alleles, but they still code for the same trait or characteristic. Now, what role does the centromere play on the chromosomes? The centromere is that restricted region on the chromosomes where it typically doesn't necessarily code for any protein, but it is a repetitive sequence that serves as the site for the docking site for the connector core, which is a protein-like disc structure that the microtubules come and bind to or aggregate to and begin the pulling apart of the chromosomes into sister chromatids. And this is important. The centromere is also important because it helps us to establish the actual morphology of the chromosomes and how they are identified. And this is where you get the different appearance or morphological structures of the chromosomes, whether you're talking about the metacentric chromosome, the submetacentric chromosome, acrocentric, or telecentric chromosome. And that centromere, based off its location, will divide the chromosome into two parts that can either be considered the short arm or the long arm. The long arm would be considered or denoted by a Q, the short arm denoted by a P. If it is a metacentric chromosome, then that means the centromere is located in the middle and it produces two equal halves. And this is what it tends to look like during anaphase and metaphase. In a submetacentric, it's between the middle and the end, so it doesn't have two equal halves. It's almost like a 3/4, 1/4 structure or shape in terms of the distance between the chromosome and the ends of the centromere. And this is what it looks like in an anaphase shape. The acrocentric is where the centromere has gone close to the end to where it produces a stalk and a bulb-like structure. It has not fully terminalized. The centromere hasn't terminalized to the end. So there's just a small piece, a satellite that is considered at the end, but it's close to the end. And it looks like this little shape here in anaphase. And this is how it looks during metaphase when it lines up at the middle of the cell. And then telecentric, think of telecentric as it terminalizes. So the centromere is at the end, it's terminalized to the end, and it only has one portion of the chromosome, which is what you see here. So with the chromosomes, you have, as we talked about before, you have somatic cells. Your somatic cells are going to be considered your body cells, and they have homologous pairs of chromosomes, okay? You have one from, as I mentioned earlier, one from your paternal and one from your maternal. And so while they're not exactly the same, they carry the same composition of genes for various traits. And so you have a total of 46 chromosomes that exist, but it's because there are 23 that occur in pairs, and that's where you get the 2N. 2N represents a diploid organism. There are some exceptions, as always, like your bacteria and viruses, they only have one chromosome. But when you're talking about human cells or multicellular organisms, they oftentimes have more than one chromosome. One thing I want to point out too is you have this homologous pair of chromosomes, and these two right here are considered sister chromatids. These are both from the paternal or the father, and this is from it's maternal chromosomes, both from the mother. So how do we identify the structure, the morphology of a cell? The chromosomes within a cell, it's called a karyotype or karyotype map. And this is a physical representation of the chromosomes within a cell. And you're able to develop a karyotype by isolating the chromosomes from a cell by treating the cell first with colchicine. Colchicine is Colchicine. Colchicine is a solution that when you treat the cells with it, it causes the cells to rapidly go to metaphase where the chromosomes are going to be in the most condensed state. And then you're able to lyse open the cells, take a picture of the chromosomes, and then digitally go in and line them up to where you are able to pair each chromosome set. And so this is how you produce a karyotype map. You'll see that this comes into play and is important when an individual undergoes genetic counseling. We'll talk about in chapter 3 and kind of go over it a little bit more in chapter 4. But when there is the thought of a potential disorder and you want to identify, like, let's say a fetus, if a woman is pregnant, she has an amniocentesis, a karyotype map can be developed. These are some of the genomes of organisms and their haploid number, such as humans, which is 23, Jimson weed 12, chimpanzee is 24, black bread mold is 8. And so you can look at a genome and look at the genetic information in a haploid set to determine what they would look like in meiosis. Now, with, as I stated, homologous chromosomes in a diploid organism suggests that the origin of that genome comes from two different beings, maternal and paternal. And so while they, again, are different, while they contain the same type of genes, they are different. One parent may give a dominant form of a gene and one may give the recessive, and this combination produces variations of traits that you may see, whether we're talking about a genetic disease or a trait that is produced. And this is what biparental inheritance comes from. It's inheritance from two parents, referring to diploid organisms. On a homologous chromosome, there is a specific site or locus where each gene is housed, and they are denoted usually by a number and a letter, whether it's on the long arm or the short arm, which will show you a P or a Q. So with the centromere is essential because it helps with determining the morphology of the chromosomes. But you'll see in some chromosomes, like your sex chromosomes, the centromere may differ significantly. The X versus the Y chromosome has a different morphological shape, and is vastly due to outside of evolutionary pressures and factors, and just them being different. The centromere location is different as well. However, both X and Y chromosomes are considered homologous, or they behave as homologs because they are both sex chromosomes, and they're essential for sex determination. So while they're not exactly the same, they do share regions that are essential or have an overlap and are necessary for crossing over, which we'll talk about shortly in sex determination. So how does the partitions in chromosomes into dividing cells work? Well, I want you to think about this. There are two types of division that occur in the cell cycle: division of the cytoplasm and division of the chromosomes. Division of the cytoplasm is important just as much as the division of the chromosomes, because if you have unequal distribution of the cytoplasm, it can result in a non-viable cell. And so that is also important. You have, so paying attention to those partitions and that process is essential into dividing cells. So a zygote is formed from the fusion of egg and sperm. So the single cell fertilized egg of multicellular organism, but it's fertilized by sperm. Okay. Karyokinesis is the genetic material partitioned into daughter cells from nuclear division, and that process is of nuclear division. So you can see here is a diagram that shows you the difference between karyokinesis and cytokinesis. Karyokinesis is the division of the nucleus. Cytokinesis is division of the cytoplasm. And karyokinesis, the nucleus of the cell is divided into two daughter nuclei, and the cytoplasm is divided in the cytoplasm of the parent is divided into two daughter cells. So ultimately, one is dividing of the nucleus and the genetic material, and the other is dividing of the cytoplasm. So let's look at more of the cell cycle. Within the cell cycle, you have various phases, one being interphase, and interphase includes multiple phases like the S phase and the gap phases. The gap phases, or G1 and G2, are important for the production of proteins that are essential for the upcoming stages or phases in the cell cycle. And the S phase is essential for the synthesis of DNA. G sub O is a point in G1 where the cells are quiescent. So they're not dividing, but they're in a metabolically active state. And sometimes you'll see cells that can stay dormant or stay in this phase for a long time, years. So what happens in interphase? And that is essential. And actually, let me go back to this part because I want to point out, if you pay attention to the timing, mitosis is a small portion compared to the overall cell cycle. Interphase is actually much longer. This is interphase than mitosis. One, it takes a long time to duplicate the genetic information, but it also takes a long time to produce all of the components, the enzymes, the proteins needed to carry out the S phase. And so they're produced in G1. So whatever proteins are needed, enzymes needed to carry out the S phase, DNA replication, are made in G1. Whatever proteins are needed to carry out mitosis are produced in G2. And in this interphase, you will have what are called checkpoints. And these checkpoints are essential because they serve as like the guardians of the cell to make sure the cell does not go into the next phase without being properly equipped and functional and viable. If it is not, as you'll see in a couple of slides, the cell will be marked for death or apoptosis. So will not be allowed to proceed to the next phase without passing that checkpoint. So as I stated, in interphase, you can look at the chromosomes, specifically the nucleus, to see what state the cell is in. Usually in interphase, the chromosomes are in a chromatin state, so they're very loose. It almost looks like spaghetti under the microscope, which lets you know that the cell's actively undergoing replication. And so you're not able to discretely identify one chromosome from another. So ultimately, in interphase, there is an absence of visible discrete chromosomes. And I thought this was just a nice representation to help you visualize how much time is spent in each of those phases in terms of hours, interphase versus mitosis. Significantly different. And even when you further go and look into mitosis, prophase is the longest part of mitosis than metaphase, anaphase, or telophase. So what happens in prophase? So in prophase, that's the first stage in mitosis. The centrioles are going to divide. They're going to move to poles, and you're going to have the nuclear membrane that is still present, but it's slowly breaking down. So that's a clear indication that you're still in prophase. There's one nucleus, and it's still present, but it's breaking down. Okay. The chromosomes are beginning to condense. They may not fully be condensed, but they're beginning to condense and become discretely visible. The sister chromatids that should show two parts, right? They're connected at the centromere and they're held together by multiple subunit proteins called cohesin. Cohesin is actually made in G1. I'm sorry, in G2. Okay, and cohesin is important because it helps hold this complex together. If these complexes are not held together, it will have degradation. Prometaphase. Prometaphase. Let me pause. Oops, let me pause this. Okay. So in prometaphase, this is the stage between prophase and metaphase. So sometimes in the beginning, depending on what textbook you read, it may be considered late prophase. So you have early prophase and late prophase. Consider prometaphase like the late prophase. And this is where the chromosomes are discretely identified one from another, and they're preparing for metaphase so that they can move to the equatorial plane or the midline or the middle of the cell for metaphase. And so at that place, you have the centrioles that are formed, the spindle fibers that are formed as well. In metaphase, this is where the chromosomes meet at the middle of the cell or the metaphasic plate, and they have aligned so that they can begin to be pulled apart. And at this place, you should also see the presence of the connector core. In the very beginning, I talked about that disc-like protein, disc-like structure that binds to the centromere. This is what I was referring to, and attached to the connector core, this disc-like structure, is where the microtubules are going to bind so that it can begin that force of pulling apart these sister chromatids from each other. Now you'll see in meiosis that how the chromosomes align at the metaphasic plate is also a level of variation that provides genetic variability. Outside of cohesin helping to hold the sister chromatids together, you have shugosin, which is another protein that actually protects cohesin from being degraded by another protein, separase. Separase degrades cohesin. And so all these proteins are necessary and they're made in G2 because they help to stabilize this structure. And once separase is necessary because once the chromosomes don't need to be held together again, separase can come in and start to degrade those proteins. And so all these proteins are essential. And as you're studying this, I want you to think about what would happen if one of these proteins was mutated and did not function. How would this affect the cell? Think about if cohesin was mutated. How would that affect mitosis? How would that affect meiosis? Anaphase and telophase. Anaphase is disjunction. Now you're going to have the pulling apart of the sister chromatids and they're going to separate into daughter chromosomes. This is important. Anaphase is an extremely important process, especially when we see this in, meiosis, because when you don't have proper pulling apart or disjunction, it can result in genetic abnormalities, which we'll talk about in meiosis. But anaphase, you can identify it because the chromosomes are still discretely identified, but they're being pulled apart to opposite poles in the cell. It's still one cell, but it's elongated, and you have polarization that is more evident. In telophase, you're going to have now a more clear formation of the preparation of cytokinesis, which is the formation of two cells. You're going to have the nuclear envelope that's starting to reform, and the spindle fibers are going to disappear. And that reforming of the nuclear membrane is preparing for cytokinesis, which is the formation of two separate cells. Okay. Once you get to the ending of telophase, cytokinesis role is to produce two new cells. Okay, in plant cells, it's a cell plate that is formed. Okay, and in animal cells, you have a cleavage furrow, which is the constriction of the cytoplasm where it kind of folds in on itself and forms two new cells. So these are some of the stages visually of the chromosomes in the nucleus during the phases of the cell cycle, whether it's interphase, prophase, prometaphase, metaphase, anaphase, and telophase. So what is happening in mitosis? You have the partitioning of chromosomes into dividing cells, and it produces daughter cells that are full diploid, so two daughter cells that are diploid. So the end of mitosis produces two daughter cells that are diploid and identical. There are mutations that arise from errors in the cell cycle, and they are vast. As we progress through, you'll learn about some various disorders. One that is very clear that we'll talk about, not necessarily mitosis, but meiosis, is Down syndrome. However, to help prevent some genetic abnormalities due to errors in this process of cell cycle, those checkpoints are essential, as you see here. And one of the components that helps with these checkpoints are cyclins and CDKs. These are proteins that come together, and the presence of these proteins help determine whether the cell can proceed to the next phase or not. If they're not properly formed together, then the cell will be halted at that phase and not allowed to pass through to the next phase. Well, I don't want you to have to memorize every single cyclin and CDK combination. You should be aware that cyclins bind with CDKs, and they are help in regulating the checkpoints in the progression of the cell cycle and regulation. This is a time that you can pause and do a check your knowledge to see if you understand this. So the question is, if an organism has a diploid number of 16, how many chromatids are visible at the end of the mitotic phase? Pause, try to answer this question on your own. And the second question is, how many chromosomes are moving to each pole during anaphase of mitosis? The first answer is 32, and the second is 16. So let's go into talking about meiosis. Now, meiosis is also known as reduction division because it reduces the amount of genetic content and chromosomes within the cell. And different from mitosis, it doesn't produce two diploid cells. It produces 4 haploid daughter cells that are genetically unique. One of the important processes in meiosis is crossing over, and this occurs in Prophase One. There are 2 rounds of meiosis. There's Meiosis I, which is the reduction portion, and meiosis, which is the equational division. But in crossing over, this is where you have the physical exchange of genetic information between homologous chromosomes, and this crossing over that occurs helps provide genetic variability and uniqueness to chromosomes. And so you can see here, you have synapses of bivalence, okay? The synapses is the site where you have the chromosomes, homologous chromosomes that are connected together. The crossing over is visibly seen when a chiasmata is formed, and that's the site where the physical exchange occurs. And as a result of crossing over, you have recombinant chromosomes that are produced. And as you can see, instead of having 2 red and 2 blue, now you have 4 different or genetically unique chromosomes. So I had to pick this back up. So if it sounds a little bit different, my speaker actually with my microphone went out. So hopefully the sound is not too significantly different. But just an overview of meiosis, you have 2 rounds. The first cycle of or round of meiosis is reductional division, and the second is equational division. So I won't go into detail about meiosis II because it really does mirror mitosis. The more unique portion of meiosis is Meiosis I. Okay. Also keep in mind that there's only 1 round of DNA synthesis, and that occurs in interphase, and it's at the beginning or before Meiosis I starts. It does not occur again in between Meiosis I and II, and that process in between Meiosis I and Meiosis II is called interkinesis. But there is no doubling of the genetic information. It does not go back to replication because it would defeat the purpose of meiosis. It will produce, again, diploid cells, and you want haploid cells. So when you compare mitosis and meiosis, you can see just very clearly that mitosis produces 2 daughter cells that are diploid. Meiosis produces 4 daughter cells that are haploid. And so the genetic information, as you can see here, is half. The first division of Meiosis I is the focus on one of the most important parts is Prophase One. There are 5 substages in Prophase One: leptonema, zygonema, pachynema, diplonema, and diakinesis. And then you have the end of Prophase where the centromeres are present in the equatorial plate. In leptonema, the chromosomes are going to look like long single threads, and they're unassociated with each other. So they're not fully condensed yet and to 2 discrete bodies, and they're forming they're preparing to become more condensed. In zygodyma, you have the synapses or the synaptical complex that it forms, and that helps to hold the pair of homologous chromosomes together. And when they're held together, that is known as the bivalent, bi meaning two. Pachynema is where the bivalent becomes shortened and thickened, so they become more condensed, and they split into two sister chromatids. And when they split into those two sister chromatids, they're now called tetrads, indicating that there are four there. This is when crossing over can begin, because now you have them discretely condensed and their synaptical complex has formed. They're closely aligned with each other. And let me also say the synaptical complex, think of it like a zipper-like complex, and it's proteins that are produced to hold them together. And this is essential in order for crossing over, which occurs during this stage or begins in this stage, to occur. And this stage, crossing over, occurs, which is the physical exchange of genetic material between non-sister chromatids. Diplonema is where you're looking at specifically within this tetrads, the sister chromatids are going to separate, and the chiasma is going to be visible. And this is where the sister chromatids are still going to be intertwined, but you clearly see that crossing over has occurred. In diakinesis, the nucleus and the nuclear envelope have broken down, and the centromeres attach to the spindle fibers as it's going to prepare to go into the next round of meiosis, which is the next phase. You have now the next stage, and I want you to just kind of visually see what is happening in metaphase, anaphase, telophase, and then going straight into prophase 2. Well, there's a small period of interkinesis, but then going into the next round of Meiosis II. You have the lining up of the homologous chromosomes at metaphase, and I want you to pay a note that the combination of chromosomes in the cells produced are important. So in metaphase, two tetrads align in the center of the cell. Each tetrad contains a blue dyad and an orange dyad. Crossover occurs between the non-sister chromatids, and then in Anaphase 1, the dyads of each tetrad are pulled to opposite poles. So remember, in anaphase, it's the pulling apart. It's that force that pulls them apart in preparation for two cells. In telophase 1, the nuclear membrane starts to form around those dyads, and the cell begins to divide. And that will then lead you to prophase 2, or Meiosis II, which you see here. In Meiosis II, the dyads are in the center, or aligned in the center of the cell, so they meet at the metaphasic plate of the cell. In Anaphase 2, the sister chromatids of each dyad are pulled to opposite poles or ends of the cell. In telophase 2, the nuclear membrane forms around each of the sister chromatids. Okay, so there's that nuclear membrane that is forming around each of the sister chromatids, and the cell begins to divide. So you're starting to have that cleavage furrow to form two cells. And now you have haploid gametes. So you have 4 haploid gametes that are now genetically unique and non-identical. So again, in the first round of division or metaphase 1, the chromosomes are at maximum shortest, means they are highly condensed, and the chiasmata terminalizes. So it's holding the sister chromatids together, allowing for the crossing over to occur, and it lets you know that crossing over has, completed when you have that terminalization of the chiasmata. Anaphase 1, very important protein is no longer valid, and that's cohesin. Cohesin has to be degraded, and that's by the help of separase, because you don't need them to be held together anymore. You want the pulling apart of these sister chromatids so that you can form 2 new cells. So cohesin should not be present during Anaphase 1. And that allows for the homologous chromosomes to separate, and non-disjunction may occur, meaning the improper separation and division of these chromosomes can occur, and that can lead to genetic disorders like Down syndrome, i.e. trisomy 21, 3 copies of chromosome 21. Cytokinesis is going to form so that you can have the equal distribution of the cytoplasm and producing 2 haploid cells. The nuclear membrane, again, is going to form, and the nuclei are going to enter into interphase or interkinesis. So just summarizing Metaphase One and or Meiosis I, you have the homologous chromosomes that separate and move towards the poles. The sister chromatids remain attached to the centromeres. The duplicated chromosomes are going to reach the opposite poles of that cell, and now you have a haploid set of chromosomes. Cytokinesis has to form because those two haploid daughter cells have now formed, and you need to form two separate cells that have their own nuclear envelope. With Meiosis I, crossing over is important, and that's what makes Meiosis I extremely important, if not the most important part of meiosis overall. And that is because that is where crossing over takes place. And without crossing over, you do not have genetically unique gametes, i.e. sex cells. So it's very, very important, okay? It's a level of genetic variation that is necessary. Meiosis II is the cytic mitotic division, and I won't go into details about Meiosis II because it resembles that of mitosis. So if you know mitosis, you know Meiosis II. Okay, so let's go into the development of gametes and how they form, whether we're talking about sperm or egg. So the process of developing gametes is referred to as spermatogenesis or oogenesis. Spermatogenesis is the production of sperm cells. Oogenesis is the production of oocytes or eggs. Males undergo spermatogenesis in the testes. Females undergo oogenesis in the ovaries. And you can see here that there are vastly different. You have at the end of spermatogenesis 4 sperm, and oogenesis you have 1 oocyte. So what happens in spermatogenesis? You have the initial spermatocyte or spermatogonium that goes through various rounds of meiosis or division. Ultimately, as it goes through that process, it produces a spermatozoa, which has matures to have a tail, and that's what gives the sperm its ability to be mobile or to move so that it can fertilize the egg. Ultimately, the sperm, which are produced at will, males are not born with the amount of sperm they're going to have. They can produce sperm for years and years, and they can produce sperm. I think it's about every 72 days or so, new sperm is produced. It takes about 70 days, give or take 60 to 70 days for sperm to be produced and to mature. But once they are mature, they have a fresh turnover of sperm about every 70 days. Eggs are much different. The formation of ova in the ovaries produce mature cells, but it is reduced because it's not 4 eggs that are produced. It's 1. It produces 1 ovum and 3 polar bodies, and this is due to unequal distribution of the cytoplasm, and in part it's due to the ovum, the egg, needing all of those extra nutrients and components because a female is born with the amount of eggs she's going to have. She does not produce new egg every 70 or so days, and so due to the lifespan of the longevity of these eggs, they need a larger amount of nutrients and components to support it and sustain it. So all of the unequal distribution that you see is for that reason, and that's why you have 1 large ovum and those small 3 polar bodies which disintegrate and do not become viable eggs. However, each egg or ovum that is produced is genetically unique. And just to point out, when you have a zygote that is formed, okay, it is formed off of the ovum. So ultimately the sperm fertilize the egg, but they only yield the genetic information. They don't give the organelles. The organelles that the zygote has come from the mother. And this is why a lot of mitochondrial diseases are maternal originated. So why is meiosis critical to the success of reproduction? 1, meiosis is the mechanism that produces genetically unique and haploid sex cells. And if this process does not happen, if there's abnormalities with it, it will result in an abnormal zygote if that is formed, okay? But just like animal cells go through this process, plants and other fungi life also have an alternate life cycle between this diploid and haploid cell, and they have a form of gametes as well. So you can see here that there is an alternation of generations between the diploid sporophyte and the haploid gametophyte. Spores, think of spores as the seeds that are waiting in a dormant stage until the environmental factors are viable for or conducive for proper growth and fertilization. So, and we're talking about a multicellular plants. So the process of meiosis and fertilization bridge the two phases of the life cycle. And angiosperms, which are your flowering plants, shown here, as you see here, the sporophyte stage is the predominant phase. Okay, or phase, I'm sorry. So the sporophyte phase is the predominant phase. So how does this work? The stages in the life cycle of this multicellular plant that you see here are divided into two stages. One as a diploid, which you see here, the predominant phase, and one as a haploid, which you see here. The stages are can quickly be summarized or defined as the flower of the diploid, which is 2N, and one haploid stage. Stages are summarized as follows. So here we go. The flower of the diploid sporophyte contains a megasporogonium. Make sure I say this right, you guys. Megasporogenium. And this produces the megaspores. These megaspores are necessary for production of future plants and offspring, but you also have the microsporangium. And this is also important because it produces the microspores. The meiosis produces haploid, megaspores, and microspores. So the end of the process of meiosis for plants should produce two different gametes, the megaspores and the microspores. The megaspore becomes the female gametophyte or embryo sac, and the microspore becomes the male gametophyte or the pollen grain. The sperm of the gametophyte fertilizes the egg of the female gametophyte to get a diploid zygote, and that's what you see here. So the female gametophyte combines with the male gametophyte, the pollen grain. They fertilize it to form a zygote, and then it's just a cycle that keeps happening again that I talked about, that alternation of generations between diploid and haploid. So let's look at a little bit of application. How can you look at these chromosomes and what do they tell you? So you can use electron microscopy to look at the visible appearances of the chromosomes. They are visible during states when they're highly condensed, and that would be mitosis and meiosis. If you were to try to look at them in any other stage, it would be very difficult to identify one chromosome from another. These chromatin fibers make up the chromosome, and they coil and condense. And I want you to also remember that this is also composed of proteins, okay? So electron microscopy of these mitotic chromosomes can be seen in various states, and they look like coiling, which is called the folded fiber model. So in these different stages, you can see the chromosomes in the various condensed stages, and it helps us identify what's happening in the cell and if there are any abnormalities. So this is the end of chapter 2, and this again is the case study questions that I want you to consider based off of the opening case study that we talked about. But as you're studying this, also think about it from a female perspective. What happens, or what would happen if it were eggs that were affected as opposed to sperm? That is chapter 2, and look forward to chapter 3.